Do microplastics exist in the deep sea, and do organisms of deep sea habitats such as hydrothermal vents actually ingest that microplastics? Is it in their system? Well, we're going to find out how that experiment's going to take place, how that study's going to take place, because we have a good friend of mine, Dr. Andrew Thaler, who is on the podcast again. If you remember correctly, he was on the podcast a couple of weeks ago talking about deep sea mining and sort of the state of that deep sea mining. But now we're talking about one of the projects that he wants to do based on some of his PhD work and sort of the a new stewardship of this new data collection of 20 years of deep sea data of organisms in the deep sea. We're going to talk about what he's going to do with that data, why it's important, and other studies that could come out of that data, of that use of that data. On this episode of the How to Protect the Ocean podcast. Let's start the show. Hey everybody, welcome back to another exciting episode of the How to Protect the Ocean podcast. I'm your host, Andrew Lewin, and this is the podcast where we find out what's happening in the ocean, what you can do to speak up for the ocean, and how you can protect the ocean or live for a better ocean by taking action. On today's episode, we have a good friend of mine. I feel like he's like family in the marine science and conservation realm for me. This is Dr. Andrew Thaler. We talk about a new study a pilot study that he is trying to crowdfund for. So we're going to put a link to that crowdfunding site so that you can help, contribute to that study. But it's going to be an important study. He's got a lot of data of deep sea organisms that are part of hydrothermal vents and very interesting deep sea habitats from various places around the world. This is data that probably spans more than $20 million in terms of getting that data, spending money on the boats, on the ROVs, on the sampling, just to get all these samples. Over 80,000 samples or over 80,000 organisms. Different species. It's a complicated data set that Andrew's become the new steward of. And he wants to do stuff with it. He wants to analyze it. He wants to, ask questions that these samples could help answer. And the one that he's going to talk about today is, do microplastics accumulate within these organisms of these very sort of highly sulfuric types of habitats? Hydrothermal vents are very complex, can be very dangerous habitats for a lot of species and other species who have adapted to this and are able to live within these areas. It's a phenomenal study. I highly recommend that if you are going to contribute to anything about science and marine science and conservation, this is the thing that you should be contributing to, the study that you should be contributing to, because it's going to answer some very interesting questions, some scary questions that I don't know if I want answered, but needs to be answered is, how many microplastics are in there and does it accumulate over time? That's something that we need to know because we need to answer these questions. We need to really take hold and understand what's happening in the deep sea. How is this affecting the deep sea? But the first thing is to get a baseline data of what microplastics or how much microplastics are in the deep sea organisms. So we're going to find that out today. Let's get started with the interview with Dr. Andrew Thaler, talking about his new study that he wants to crowdfund for, or that he is crowdfunding for. I'm going to put a link in the show notes, so feel free to click on that and contribute if you can. But here's the interview with Andrew Thaler. Enjoy, and I will talk to you after. Hey, Andrew, welcome back to the How to Protect the Ocean podcast. Are you ready to talk about your science experiment coming up?
Absolutely, and thank you for having me.
No problem. You know what? I always love having you on. You were on actually just a couple of weeks ago, maybe longer than that. It feels like a couple of weeks ago. You talked about deep sea mining and everything that was going around that. And during that interview, we actually got to talk about what you're up to next. And I always laugh when I always say, "Hey, what are you up to next?" Because normally when I ask people, they say one thing. You have like a plethora of things. You just kind of like, "Hey, I've got this, I've got that." And one of the things that you mentioned was you had these data samples that you wanted to examine for microplastics, and they're deep sea samples, which we'll get into in a little bit. But I just love it because, I feel like the biggest tragedy in science is when you have samples of a certain data range and nobody uses them for something, or they're just sitting on a desk being like, "Oh, I want to use that for this idea that I have or this question that I want to answer." And so you've taken on sort of, you inherited these samples, which we're going to talk about today, and talk about this entire experiment that you're about to do, the study that you're about to do. And with that, we are going to ask the people at the end, but I want to do it now so that people can take a look as they're listening. But if you go to experiment.com, Andrew, you're fundraising to help you to fund the actual experiment. Because this is something that you're doing on your own with your own sort of time, and it's something that you're going to have to put a lot of work in, and there's always money that's involved in that kind of stuff. So we're going to talk about that. I want to mention it at the beginning because it's experiment.com, and then you go look for Andrew Thaler, and I'll put a link right in the show notes so people can go and just kind of go. If you want to participate in funding that or partly funding that, you can go ahead right now, and you can do that as we're listening. But anyway, Andrew, why don't you just kind of remind everybody before we get into all that experiment stuff, why don't you just remind everybody who you are and all the things you do, and briefly, but all the things you do.
All right. Well, thank you for that. I'm Andrew Thaler. I am a deep sea ecologist, conservation technologist, and an ocean educator. I do a lot of work around human impacts in the deep sea, particularly around chemosynthetic ecosystems like hydrothermal vents and methane seeps. I also do a lot of conservation technology projects, building ocean sensors. I'm apparently involved in politics. We talked about that a little bit on the
Yes,
last
we
episode.
did. Yes, we did.
Um, And so today I want to talk about a project that is just kicking up, establishing a baseline for microplastic accumulation in deep sea animals using a historical sample archive. It is a mouthful of a title for a project, but that is what it is on experiment.com, where you can go and find it. And one of the
It's your marketing genius of that title.
absolutely, you are guaranteed to find it if you use 3 or 4 of those keywords. Um, But so we are crowdfunding this project. It's a little bit of a throwback to the glory days of science communication online
Yeah,
when we were doing big crowdfunding campaigns. So it's been fun to sort of dive back into that sort of a fundraising model. It's a little bit different because we have support from the Experiment Foundation. So they've matched about 2/3 of the funding,
that's
which
awesome.
is nice because it means that we're pretty close to our goal just right
Yeah.
off the bat, and also it means that any dollar you donate is going to be matched not once, not twice, but thrice.
Oh, that's awesome.
So that is a really really nice thing to have. But I want to start with a little bit of a story, and I've been trying to
Yes.
think of the best way to describe how I came about this archive of biological samples. Because I really want to convey sort of the importance of carrying on science from samples that have already been collected. So, I've been saying that I've inherited them, or that I've taken ownership of them, or that I'm the
Mm-hmm.
custodian of them. But I think really what's happened is that I am the new steward for a collection of biological samples. My PhD advisor is in the process of winding down her lab and getting ready to retire, and contacted me and said, "Hey, we have a 20-year archive of biological samples, and these are a lot of samples from my own PhD research out in Papua New Guinea, samples from the Mid-Cayman Trough, the deepest hydrothermal
Ooh,
vents in the world,
wow.
methane seeps in the Gulf of Mexico and on the Atlantic margin, including places that will soon be marine national sanctuaries, which
There
is
we
a
go.
very exciting,
Nice.
exciting process." Um, and they've been collected over the course of the last 2 decades. And, one of the unfortunate things that happens when a research lab winds down is that we've all taken tons of samples, and it's not always clear where those samples end up once your science is done. These are all secondary samples. The primary science that they were collected for has been completed, the research has been analyzed, the data has been published. And so now we literally have buckets of samples just waiting for new projects. And one of the things I've always been interested in understanding is we don't really know where most of the plastics that have entered the ocean have gone.
Mm-hmm.
We have these big dramatic maps of ocean gyres and big
Mm-hmm.
floating piles of trash, and we're talking about islands the size of Texas, which
Mm-hmm.
aren't real but is evocative enough to give you an idea of what's going on out there. And that's really just the absolute tip of the iceberg. Something like
Wait, but
nine.
wasn't there a project that cleaned it all up though? That said they were going to clean it up.
I, you know,
There's somebody said they were going to clean it up in like 5 years. I believe.
Big promises, little payoff on that one.
Sorry, that was a bit of a jab at that other project that you had nothing to do with. Nobody had anything to do with that. We know, but yeah.
And most of this plastic, 90%, 95% of it is sinking into the deep sea. It's breaking down at the surface. It's being photodegraded by sunlight, and it's sinking into the deep ocean. And we don't know where it goes, or what happens to it, or how it gets incorporated into food webs,
Right.
or how it is aggregating inside animal tissue. There's been some recent research at cold seep that shows that the animals that live in cold seep sediment now have microplastics inside their bodies, which, just to give you
Wow.
a perspective of the sense of time here, we discovered methane cold seep ecosystems in 1983.
Okay.
So if you were born before 1983, you are older than human knowledge about this specific ecosystem. And we are already finding plastic trash in ecosystems we barely knew existed, accumulating up the food web inside of these ecosystems. And we don't really know what it does. We don't
Right.
know what the detrimental effects are going to be. We don't know what the real harms are. We have a very poor understanding of the deep sea in general, and an even
Right.
poorer understanding of how plastic is impacting the deep sea. So one of the things I wanted to do with these archival samples, secondary samples that have been processed for the research they were collected for, and so one of the things that means is that they're not in the world's greatest condition. They've already been sampled and subsampled. They've been, in some cases, sitting in buckets of literal 5-gallon buckets of ethanol
Right.
for 15, 20 years now. These aren't museum quality specimens that you would put on display, but that makes them perfect for something like a microplastic study, because in order to find out what microplastics are doing inside the tissues of animals, you have to dissolve the animals.
Right.
And so it's a very destructive process of doing those studies. And so a sample set like this, where we feel comfortable using these organisms for that kind of destructive sampling, makes it really, really enticing to do something like understanding microplastics. In addition to that, we now have a biological sample archive from the Western Pacific, the Eastern Pacific, the
Yeah.
Atlantic margin, the Gulf of Mexico, the Caribbean. This is a globally distributed sample set from hydrothermal vents and methane seeps from around the world over the course of 20 years, which means we can use it to do a baseline analysis. And what a baseline analysis means is that because we know almost nothing about where the plastic is going in the marine environment, we just need to take this initial sample set and find out what the level is from this decade and last decade, so that we can begin comparing it against other archival samples if we have them, and
Hmm.
against new samples going forward into the future, to really provide that baseline that we can ground truth any new studies out of. Because let's say in 50 years someone goes to a hydrothermal vent and brings back a bunch of shrimp, and the shrimp are full of plastic in their tissues.
Yeah.
We need to know what it means that they're full of plastic in their tissues now compared to 20 years ago, compared to 30 years ago. Having that ability to make those comparisons lets us say something about, is plastic building up more and more over time? Is it dissipating into the marine environment? Is it being broken down? Chemosynthetic ecosystems are some of the toughest ecosystems on the planet. These are
Right.
animals that eat hydrogen sulfide and poop nothing because they
Yeah.
don't have digestive systems.
Yeah.
Um, they have evolved to live in an incredibly toxic environment that most other animals on the planet would not survive in. They
Right.
can thrive at 80 degrees C, which is
Yeah.
frankly astounding. Um, and so, the question is out there. Are microplastics accumulating in these ecosystems? To what extent? And what can we learn about microplastic uptake in the deep sea by looking at this archive of historic samples that I now have stewardship over and a
It's
responsibility to take in the next generation?
Yeah, no, absolutely. And I mean, it's definitely an important study to do, right? If you think about the data range, just the date range of the data, 20 years is impressive.
Hmm.
Uh, you have the geographical expansion. You're covering a lot of the world oceans, or parts of the world oceans, which is great. they're definitely interesting habitats, to say the least, in terms of how they function, and definitely brings, you kind of hit the nail on the head in terms of these can be very difficult environments for to live in. So the animals that live in there are in there for a reason. It'd be interesting to see if it has an impact, and getting at baseline. Even like having it through the date range, you might be able to get would you be able to get, you think you'll be able to get a sense between that 20-year, like, obviously they're probably labeled what year they were, collected in. Do you have data, say, from like Papua New Guinea from like 2000 to 2020, or is it just more of, are they all sort of when that study happened, like during your PhD, that was like the 3 or 4 years that you were there, and then another, like the one in the Caymans is another, maybe 5 or 6 years. Like, it's not all, you're not getting like Papua New Guinea wasn't from 2020 or 2000 to 2020, right?
So for some of the sampling sites, we do have multiple samples for multiple times, and
Okay.
Papua New Guinea would be the prime example of that because that was a proposed mining site, and so we went back several
Right.
times, and we have about a decade of sampling
Okay,
from
that's
Sol
good.
R1 in Papua New Guinea. But for other sites, and one of the other reasons why making sure that samples that have been collected get maximally used, some of these sites we will never visit again.
Right.
There are discrete methane seeps that we will almost certainly never send an ROV back to because the
Right.
ocean is so big and there's still so much to
Yeah.
discover. And it is so expensive to mount a research expedition that novelty really drives a lot
Mm-hmm.
of the prioritization of sites.
No
Um,
doubt.
the Cayman Trough, we have several research cruises from. There was about a decade where the Mid Cayman Spreading Center was the best studied hydrothermal vent system in the world because we sent like 7 research cruises to it over the span of a decade, which gives you a sense of
Yeah.
how little it takes to actually do intensive, what counts as intensive studies of the deep sea. Um, but for some of these sites, some of the smaller methane seep sites, some of the hydrothermal vents on the East Pacific Rise, some of them don't exist anymore because a hydrothermal
Mm-hmm.
vent is also an active volcano, and volcanoes do what volcanoes do, and then there's no vents there anymore.
Right.
Um,
Right.
so, it's a snapshot in time, and it's an archive of some places that we may never see again. And even then, we're going to be finding out. And I have hypotheses about what we're going to discover, and I'm fairly certain we're going to be finding microplastic at most of these sites. How and what species tend to uptake the microplastics are going to be the big question for me. But, we're talking about the most remote ecosystems on the planet,
Mm-hmm.
places that um, we'll never see again, places that we've barely known about, and we're looking at, how significantly human activities on land have impacted
Yeah.
everything about them.
Well, it's going to be it's pretty much a scary thought with the results that, the hypotheses that you have, just seeing the presence of microplastics at so many deep sea places.
Yeah.
I think that's the scary thing. I know, we've talked about seeing plastic bags at the depths of the Mariana Trench, and plastic at but to see,
Every research cruise I've ever been on,
yeah.
every ROV dive I've ever done, we've found some form of plastic trash.
See, that's scary. That's, because I think when, before, hearing that, oh, we saw a plastic bag at the depths of the Mariana Trench, which was scary enough. Now to think about how much plastic's in the ocean, we used to just think it's on the surface, or not just think, but we used to just talk about it as it's on the surface, but we never really talked about what's below and how it's affecting animals or the habitats itself. So this one is more of, hey, is there a presence, and how much is it accumulating over
Right.
this date range? Is that the idea?
Yeah, that's the idea. And we call that a baseline because
Right.
we're starting from the zeroth principle. We don't know what normal is in this context. And so
Right.
we need to create that first data point for comparisons to be built off of.
And even the one with the Papua New Guinea, where you have a decade of data, um, would you be able? Do you think there'll be an accumulation within like that time frame? Is that
So,
one of your sort of
so that
ideas?
site's interesting because it's the closest to shore and the
Right.
closest to human habitation. So one of the reasons it was proposed as a mining site is because it's only 20 kilometers from shore and it's
Oh, yeah.
inside the Bismarck Sea. So it's in a contained chunk of ocean. I'm expecting we're going to see pretty significant
Yeah.
microplastic uptake in that ecosystem specifically. The fascinating question for me is, so you have these snails that live at the hydrothermal vents that are eating the hydrothermal vent fluid. Those are the ones hosting the chemoautotrophic microbes, harnessing the chemical energy of the vent, turning it into food. Those are the ones that don't have digestive systems. They have just
Right.
specialized chambers in their gills for bacteria to grow on. And so the question for me is, are they going to start showing microplastic uptake? Are we going to start seeing the microplastic uptake in the grazing animals that kind of live
Hmm.
around the vents and are sort of thriving off this foundational habitat that the snails are building? Are we going to see it in what we call the halofauna, which is kind of generic deep background scavengers that
Right.
aren't necessarily hydrothermal vent associated, but aggregate around hydrothermal vents because they are oases of life on the deep sea floor. So, what kinds of organisms are going to be picking up microplastics into their tissues versus not because of their feeding style or their life history? And I think we'll probably see a quite substantial variation depending on where we look. And I
Yeah.
will tell you this: the very first organism we're picking is a limpet that lives on top of those snails because that seems to me like the best target. It's right at the vent
Yeah.
interface. It's on top of the chemosynthetic snails, but it's a grazer, so it's eating all the bacteria and detritus growing on the snail shell.
So if there's anything in there, it's going to be in there. Now, when you look for microplastics, I'm thinking these tiny pieces of shards of plastic. Is that what you're looking at? Or are you looking at the chemical makeup of what goes into a microplastic? Like how, like, is it an actual piece that gets up, that gets kind of taken in?
Yeah.
Or is it just the chemical composition? Like you're looking for the,
So that's
metals or
the.
anything
I mean,
else?
that's the same thing.
Okay.
I mean, a microplastic, a plastic is a hydrocarbon chain. You know.
Yeah,
Um,
true, true. But like, are you actually
so
finding like actual pieces?
yes. So what
Okay.
we are doing is we're basically dissolving either the whole organism or dissecting out specific organs and then
Okay.
dissolving them in a very strong base. So all the organic matter is removed and the inorganic matter remains. And then we're using a fluorescent microscope to excite the plastic particles. And
Ah.
so from there, we can skim the plastic particles out. Do a particle count is usually what you do at the beginning. If you want to start getting very precise,
Mm-hmm.
and if there's enough of them, we can start taking the mass of microplastic particles removed and doing it, percent body weight
Right.
microplastic.
Right.
But we are identifying them under a microscope. And it's really funny because I'm getting a crash course in microplastics handling and lab practice because I'm originally a geneticist, so
Right.
I have very good clean room protocols, but like some of the fun things that are happening is one of the ways to make sure that things aren't getting contaminated is the clothes you use during the study. You dye with fluorescent dyes so that you can immediately identify if like a fiber from your lab coat has gotten into your sample.
Uh,
So everyone working in the lab is going to have some different crazy tie-dyed lab coat that they have to wear so that we can tell who's getting what contaminated. Which I just
that's
think is is
that's kind
is
of cool in
is, is
itself.
fun. Yeah, a
Yeah,
little
yeah.
yeah,
Well, I mean,
little bit of end of the world humor there.
but I mean, it's important, right? Because
Yeah,
it's you got to be able to identify that. You don't want to misidentify some
exactly.
thread or something that's in the organism.
Yeah. Well, and you
Um,
want to know because I mean, especially me, I work with a lot of plastics because
yes.
one of my conservation technology projects is heavily involved in 3D printing.
Right.
And
Right.
so like, my office is swimming in microplastics. These samples can't go anywhere near my office.
No.
Because I can guarantee you start looking in my tissue, I'm sure you'll find PLA everywhere.
You probably have all those the big round sort of what do you call them, the coils of all the plastic, like the 3D printing kind of
We call
plastic
that
that you
poop.
put
It's
in.
3D printer poop.
Poop, 3D printer poop. I love that. I love that. Yeah, you also have a lot
Yeah,
of 3D printer poop around, just to be able to do all the stuff that you do, which is probably a subject matter for 10 other podcasts. So,
absolutely.
which will be fun to do. So when you look at this whole project, how long will it take from beginning to end when you to get the results?
Pilot study that we're trying to fund now, we're proposing it to run through 2023.
Okay.
And that will be kind of a first look. We're going to pick, depending on how fast the protocols go and how much microplastics we identify and where kind of our lines of inquiry lead, will determine how many organisms we pick and from how many sites. But the idea is for the first pilot study, which is what we're trying to fund with this experiment project, is going to be relatively small. 5, maybe 10 species from 1 or 2 different kinds of ecosystems, in order
Right.
to start getting a handle on this. If that proves to be something worth pursuing long term. I mean, this biological archive I have is a career's worth of samples. I mean, I could be doing this for
Wow.
another 50 years and not work my way through these samples. There's probably 80,000 organisms in there. Um,
That's a
so
lot.
yeah, so one of the other things I'm working on is working with colleagues and collaborators to find out, who else has interesting research questions that they want to do that could benefit from this kind of a deep archive.
Yeah.
And so I've been, in consultation with graduate students in particular, because graduate students notoriously don't have tons of access to lots and lots and lots of samples yet.
Especially from the deep sea.
Exactly. Well, deep sea samples are precious. They're
Yes.
precious, and they're
Yeah.
they're so rare, and to have this many of them, it really is a stewardship mandate more than anything.
Right.
To make sure that they're taken care of, and to make sure that they're used, and to make sure that we continue to learn from them for as long as possible, because these organisms have given us, themselves as an opportunity to
Yeah.
understand our world, and so it's really kind of an insult to the deep sea not to use that
100%.
to its complete potential.
Mm-hmm.
Um,
So
yeah,
you
sorry,
mentioned
I'm getting a little verklempt there. I'm
no,
verklempting
no,
myself.
I appreciate that. I think that's it's really knowing that how many samples there are and how many species there are, and it's not just for one person in one study.
Yeah.
A lot of times people think about like a huge data range, but we don't really understand how many samples there actually are. You did mention that you have collaborators and people that are going to help you out with the pilot study. Do you want to say who those are? Like,
Yeah, so we're working with the University of Maryland Horn Point Lab,
okay,
University of Maryland Center for Environmental Studies, and it's Jamie Pearson's lab is
okay,
our partner in this.
perfect, perfect. And so you'll be using their lab as the lab setting.
Yeah, so they just got a funding to get the microscopes. So they
Beautiful.
have the fancy fluorescent microscopes that we'll be using.
Awesome. That's
Yeah.
fantastic.
And then, of course, whenever I do any project involving samples from Papua New Guinea, I include my colleagues from Papua New Guinea. So we
Of
have
course.
some collaborators and some students from PNG who will be working on this with us as well.
Fantastic. Love to hear that. Now, with when you get the results, how are you gonna like what do you want people to know about that? Like, what is the obviously we know the purpose is is there accumulation and what's the baseline? This is gonna be a pretty big deal when it comes out.
I
Once you
hope
get
so.
those results, right? Like,
Actually, honestly, I hope it's not a big deal.
yeah,
I mean,
true,
wouldn't it be amazing
true.
if the result
There's like
is,
nothing.
hey, there aren't microplastics floating around
Yeah,
hydrothermal vents? Man, that would be fantastic. That would be a piece of negative data I would love to publish.
that would be awesome.
I
Yeah.
don't think that will be the case, but dare to dream. Knock on wood.
Oh, for sure. Yeah. But I mean, when these results do come out, like, would you partner with other organizations who are, have a good reach when when you deal with microplastics and plastics to get that word out there, not just in the scientific journal aspect of it, but also just to the public itself?
I'm a big time science communication advocate. I mean,
Absolutely.
absolutely. I will say I have no idea what science communication is going to look like at the end of 2023. So I'm not going to commit
It
to any
might be an
strategy
AI
there.
machine for all we know.
I mean, everything could be Chat GPT. We could all be on ham radios like
Yeah.
breaker breaker. This is mud duck on the ones and twos. So whatever tool we're using to
Mud
spread the
duck. I
word
love
is the
it.
tool I'll be using.
That's fantastic. I mean, I'd love to hear that because I think it's important to get it out there. One question that I have, when you think about that, what people will ask is, will people want to know, what type of microplastic? Can you tell if it's like a microplastic from like a plastic water bottle or like a cap or like a Zippo light or a plastic light or something? Are you able to from the data, knowing the condition that it's in and how it's been sort of processed through animals and stuff like that, would you be able to tell if you did more experiments, the sort of the source?
With the pilot study, likely not.
Right.
As we get further down the line, and if we have access to significantly more funding, there are possibilities to do chemical analyses on these microplastics. But as you said, in some cases, they're
It'd be
so
hard.
far degraded that,
Yeah.
we'll be able to get down to the chemical constituents. Is it PVA? Is it PLA? Is
Yeah.
it another kind of plastic? But I don't know if we'd be able to track it to the source from.
Right.
I mean, again, we're talking about minuscule fragments that are found inside an animal's tissue. These aren't
Right.
like large blocks floating in the ocean.
Yeah.
These are,
And they're not large animals either. They're
yeah,
not.
no, these are, I mean, these limpets are tiny. They're tiny
Yeah.
guys.
Yeah. It's really
Um,
great. If you go to the link that we'll put in the show notes, there's a picture or video of those limpets, and they're pretty cool. They almost look like pennies,
yeah.
with some designs on them. But they're very interesting animals to look at if you really think about it and think about where they came from for hydrothermal vents.
Yeah.
These things are tough sons of a gun. So
That would be iron oxidizing on their shells, gives them that
yeah,
nice rust color.
gets that nice rust color. Yeah, yeah, which is insane to think about. Now, is there anything else you'd like people to know before we start talking about the fundraising about these experiments?
No, I think that pretty well
Okay,
covers the project itself.
perfect.
Yeah.
So, if people want to fund, so what are they funding when they go and they donate to this experiment?
So when you donate to the experiment, and it's on experiment.com, and again, the title is Establishing a Baseline for Microplastic Accumulations in Deep Sea Animals Using an Historical Archive Sample Archive. Um, a lot of what you're funding, some of the funding is going towards consumables, facility usages fees. I also have the open access publication fee built in there because, of course, anything I publish is going to be freely available to the public.
Right,
I don't like putting anything behind a paywall.
right.
Um, but to be honest, most of what you're funding is technician hours, the
Yeah,
time it
the
takes to
time it
do these
takes.
kinds of studies. The materials aren't particularly expensive. The expensive part's done. The samples were collected.
Right,
And I think at one point we calculated the cost to acquire for all the samples that we loaded into my little Ford Escape to drive it up from North Carolina, and I'm pretty sure it was in the 22 million dollar neighborhood,
I, I would
just in
imagine. I
terms
was
of the
gonna say.
number of research cruises that went out to collect these animals.
Yeah, it's and like the depth that they go to, the instruments that they
Yeah,
had to have, and everything. It's yeah, I wouldn't be surprised. I was going to put it above 20 million
yeah.
for sure. So think about that, like $20 million worth of samples not going to waste, being able to be used probably, hopefully, for a number of different studies to be able to answer a lot more questions that we didn't know. Are you excited to dive into microplastics, like this sort of area? Is this something that's a little bit new to you, in terms of what you've been studying in the past? Still deep sea, still hydrothermal vents, but the microplastic part's a little bit new. Are you excited to dive into this?
Oh my God, I'm thrilled. I have spent a lot of time over the last 10 years creating microplastics as a
Right.
conservation technologist that runs a 3D printing facility. And we've put a lot of work into keeping the plastics we produce out of the waste stream.
Mm,
And so I've been thinking about plastics for a long time. And so this is really my first opportunity to really get into the ecosystem functions, ecosystem effects of microplastics as it relates to the hydrothermal vents and methane seeps that have been my passion and my love for many
right.
years.
Well, this is fantastic. I'm glad you're doing this. And so, if people want to contribute to the study, you can do so in the link in the show notes. Go over there and let us know once you do, because we'd love to hear from you and why you did it. Because
Absolutely.
I think it's a cool thing. I would like to see more people do this. So that's going to be fantastic. So Andrew, again, thank you for doing this. Thank you for coming on to tell us about it. Good luck with the fundraising. I'm sure it's going to get produced with this audience especially. And we can't wait to have you back on to talk about other things you're doing, but also the results of the pilot study and to see where we go from there. That'll be awesome.
Absolutely. Thank you so much.
Thank you, Andrew, for joining us on the How to Protect the Ocean podcast. Really love having you back. It's such a great study, if you think about it. You have 80,000 samples of species where you may or may not have microplastics within them, and you are going to study them. Obviously, Andrew mentioned that he won't be able to go through all of them. It's a career study, but he'll be able to do a good portion of them and be able to find other, through collaborations and through other scientists, be able to find other questions that they can answer using this deep sea knowledge. As he mentioned, it's about $22 million worth of data, and it'd be very disappointing and almost a bit of like a bit rude to let all these samples go to waste and not be able to come up with another study for them. So the fact that Andrew has them, and that means they're in good hands. I love it. I love the fact that, he's got a lot of questions, use of technology around these studies to be able to answer the questions that he has and that collaborators may have. And I have no doubt in my mind that he's going to have some great studies that come from that. And the results of those and the description of those will be on this podcast. I promise you. There's no way I'm going to let Andrew get away with not putting it on. Not that he wouldn't want to, but not putting it on this podcast as several episodes. So we're going to hear from him very shortly after this pilot project. And I'm sure there's a lot of other things that I can talk to Andrew about. It's very easy to talk to him. He's always got his hand in something that is innovative and just fun to talk about. And so we're all always up for having Andrew on the podcast. But that is it for today's episode. I'd love to hear what you think of this data range. What kind of questions do you think you would have about deep sea data and what you think you would like to do with them? You can DM me on Instagram at How to Protect the Ocean, all one word, at How to Protect the Ocean, or you can go to speakupforblue.com/contact, and you can just kind of fill out the form. It goes right to my email once you submit it, and I will answer back as soon as I can. All right. That's it for today's episode. Thank you so much for joining us on the How to Protect the Ocean podcast. I'm your host, Andrew Lewin. Have a great day. We'll talk to you next time, and happy conservation.